Hello students, hope all of you are fine. So dear children, we are doing the lesson plant growth and development. And today we'll continue the topic growth, okay, and we'll discuss law of compound interest, that is exponential growth. So let's discuss about the law of compound interest. Growth is dependent on three factors: initial size W0, rate of growth R, and the time interval for which the rate of growth can be retained. It is just like depositing money in a bank. The money will grow at the compound interest. Growth will depend upon the initial size, that is amount of the money deposited, rate of the growth, that is rate of the interest, and the period for which it is sustained, that is period of the time in the bank. So we have the formula, that is W1 is equal to W0 E raised to the power RT. Okay, here W1 is the final size, W0 is the initial size, R is the growth rate, T is the time for growth, while E is the base of natural logarithm. The magnitude of R, or rate of the growth, has been called efficiency index by the Blackman in 1919. As the organ and organism with higher R value will outperform other with low R value. Next we have is the quantitative comparison between the growth of various systems can be made by measuring their absolute and relative growth rate. Next we have absolute growth rate, growth curve. Absolute growth rate, absolute growth curve is actual growth curve obtained by plotting growth against time. It is commonly S-shaped. Absolute growth rate is total by growth per unit time. A graph plotted for absolute growth rate for various time of grand period of growth appears bell-shaped. You can see in the diagram the peak is formed when the growth rate is the fastest. The period of increasing growth is depicted by the first part of the curve, while the period of decreasing growth rate is shown by the second part of the curve. Relative growth rate is equal to growth in a given time period by moment at start of the time period. It is growth per unit time per unit initial growth. Suppose two leaves have grown by 5 centimeter square in one day. Initial size of leaf A was 10 centimeter square, while that of the leaf B was 15 centimeter square. Though their absolute growth is the same, relative rate of the growth is faster in leaf A because of initial small size, that is, it decreases with time. So here we can compare the size of the two leaves, okay, where the increase is going on. Okay, now conditions for growth. Condition for growth. Growth involves synthesis of more protoplasm, cell division, cell enlargement, and cell differentiation. It is therefore influenced by all those factors which influence the biosynthetic machinery, availability of water, oxygen, optimum temperature, optimum light, minerals, and absence of stress condition. Okay, so first is the nutrients. They are raw material for synthesis of protoplasm as well as source of energy. It is seen that rate of growth is proportional to the size of the bulb, tuber, or rhizome, etc. It is called law of mass growth. Nutrients should be rich in the nitrogenous component for increased synthesis of protoplasm, and carbohydrate for energy and cell wall synthesis. All type of micronutrients, that is micro essential elements, and the macronutrients, macro essential elements, must be available for proper growth. Water is required for cell elongation, maintenance of turgidity of the growth cells, and providing medium for enzyme action. Even slight deficiency of water reduce growth. It may, however, promote differentiation. Water stress completely stop the growth. Oxygen is essential for aerobic respiration, hence availability of energy for the biosynthetic activity. Light is required for tissue differentiation, synthesis of photosynthetic pigment, and photosynthesis in its absence result in the etiolation. Light also influence certain stage of the growth. The phenomenon is called the photoperiodism. Next we have temperature of 28 to 30 degrees Celsius is optimum for proper growth in many plants. Higher temperature above 45 degrees Celsius hinder the growth due to excessive transpiration, then uh, denaturation of the enzymes, okay, and coagulation of the protoplasm. Low temperature inactivates the enzyme as well as increased density of the protoplasm. Gravity, vector of the gravity determines the direction of the shoot and the root growth. Direction of the light also determine the orientation of the leaf shoots. Other factors, excess of salt, minerals, deficiency, and other stress factor have a detrimental effect on the growth of the plant. Now next we will discuss the differentiation, dedifferentiation, and redifferentiation. Differentiation, dedifferentiation, and redifferentiation. Okay, so among that, first we will discuss what is differentiation. Growth is invariably associated with the differentiation. For example, when a seed germinates, it does not simply increase in size but forms a seedling. Differentiation is a permanent, localized quality, quantitative change in the size, biochemistry, structure, and function of the cell, tissue, or organ. For example, fiber, vessel, trachea, sheep tubes, mesophyll cells, leaf, etc. Okay, they are formed by the process of differentiation. The exact trigger for differentiation is not known. All the cells of an individual have the same genetic information. They are influenced by similar external factors depending upon the location inside the plant. An internal cellular mechanism, same genes are repressed, not allowed to express their effect, while other are allowed to show their effects. This causes the cell to behave in a particular fashion during the growth and after maturation. Enlargement, lignocellulosic wall thickening and emptying in case of the tracheids, widening, some enlargement, wall thickening, emptying, and loss of the end wall in case of the vessel elements, loss of nucleus, vacuolization and perforation of end wall in the seed tube members, development of abundant chloroplast in the mesophyll cells, deposition of the suberin in cell wall, tannin in the protoplast, and then death of the cork cell, deposition of silica in epidermal cell of the grasses, differential wall thickening, small vacuole formation of few chloroplast and small size in the guard cells, free nuclear division, a central canal, and secretion of the latex in the laticifers, secretion of mucilage in the root cap, elongation, thickening, and emptying of the sclerenchyma fiber, development of uneven pactocellulosic thickening in the collenchyma, cutinization of trichomes for preventing transpiration and formation of the stationary air layer, and development of caesogenous interspace to form parenchyma in the aquatic plants. Not only the plant are open-ended, their differentiation is also open. The same apical meristem cell give rise to different type of cell: xylem, phloem, parenchyma, sclerenchyma, okay, collenchyma, etc. The reason for formation of different type of cell and the tissue from the same type of tissue is called commitment or determination. It is generally due to localization and reception of particular signals. For example, cells distal to the root apical meristem form root cap, while on the periphery they form epiblema, followed by cortex, endodermis, pericycle, vascular tissue, etc. Now, next we have is dedifferentiation and redifferentiation. The process of specialization of differentiated cells so that they become undifferentiated and able to divide is known as dedifferentiation. It involves activation of certain genes which do not reverse differentiation but also stimulate cell division. Cork cambium, wound, uh, wound cambium and the interfascicular vascular cambium are always produced through the dedifferentiation. Cell culture experiments are based on the dedifferentiation of the cell and formation of mass of undifferentiated cell called the callus in case of the tissue culture. Normally, cells produced by dedifferentiation cell mature and form redifferentiated cell. That is, the secondary xylem elements, secondary phloem elements, cork cells. Now, development. Development is the sequence of the event that occur in the life history of the cell, organ, or the organisms, which include seed germination, growth, differentiation, maturation, flowerings, seed formation, and senescence. The term development is also applied to the change in the phases of the life, vegetative to flowering, leaf initiation to leaf expansion. Development occur even at the subcellular level. For example, appearance of the chloroplast in the cell exposed to the light. Last phase of development is senescence. Senescence or the old age lead to the death. The sequence of events occurring during the development of cell of the higher plant are as follows. So you can see here the sequence. Cell division, produce meristematic cell. From meristematic cell there is new cell, okay, and new cell will undergo expansion, enlargement, differentiation, then maturation will be there, and after maturation the mature cell will be produced. Mature cell will undergo senescence, that is aging, and due to aging there will be death of the individual. Now, different structures develop in different phase of the growth as well as in the response to the environment. The ability to change under the influence of internal or external stimuli is called the plasticity. The intrinsic plasticity is found in the juvenile stage of many plants. Okay, for example, cotton, coriander, larkspurs, ivy. Okay, environmental plasticity is best seen in the emergence hydrophytes like the buttercup, that is called Ranunculus flabellaris. Okay, in both cases plants show heterophyly and number of other morphological features. Heterophyly is the occurrence of different type of leaves, okay, on the same plant habitually in different growth phase or under different environmental condition. In case of environmental plasticity shown by aquatic buttercup, that is Ranunculus flagellaris, the submerged leaves are highly desiccated while the immersed leaves are broad and lobed. In larkspur, the juvenile leaves are broadly lobed while the mature leaves become pinnately divided with lobes becoming linear in the region of the flower. In Hedera helix, okay, that is called the ivy, the juvenile plant is root climber having alternate palmately lobed leaves while the adult plant has bushy habitat with opposite entire leaves. The flower, it bears flower and fruits. The fruit contains seeds for formation of new plants. Annual, biennials, and perennial monocarpic plant become senescent after the formation of fruit and hence die. Perennial polycarpic plants continue to grow indefinitely and bear flowers and fruit annually after attaining the maturation. However, growth of the perennial plant is not uniform throughout the year. In a calendar year, it shows period of active vegetative growth, flowering, fruiting, senescence, and dormancy. The different aspect of appearance of the plant in different seasons of the year is called phenology. Okay. The development includes growth and differentiation. It under control of both intrinsic and extrinsic factor. The intrinsic factor include the genetic factor and the growth regulators. Extrinsic factor are light, temperature, water, oxygen and nutrition. In your diagram you can see heterophyly in the larkspur and buttercup. Okay. Now let's discuss plant growth regulators. Plant growth regulators are small simple molecules of diverse chemical composition which in low concentration regulate growth, differentiation and development by promoting or inhibiting the same. One type of plant growth regulator are plant hormones or phytohormones. Technically, a plant hormone is a chemical substance other than nutrient produced naturally in the plant which may be translocated to other region for regulating one or more physiological reaction when present in low concentration. Five types of phytohormones are known. They are indole compound, that is indole acetic acid or IAA, adenine derivative, that is furfuryl, aminopurine, kinetin, cytokinin, derivative of carotenoids and the fatty acid, that is abscisic acid ABA, then terpenes, that is gibberellin, GA3 or gibberellic acid, and gases like ethylene, that is C2H4. Other related growth regulator are salicylic acid, jasmonic acid and brasinosteroids. Some vitamins also regulate plant growth. The plant growth regulators are broadly divided into two groups, that is plant growth promoter and plant growth inhibitor. Plant growth promoter perform growth promoting activities like cell division, cell enlargement, pattern formation, trophic growth, then flowering, fruiting and seed formation. They are three in number: auxin, gibberellin and cytokinin. Plant growth inhibitors normally induce dormancy and abscission. They have, however, an important function of inducing the plant response to wounding, biotic and biotic stress. Abscisic acid is known as plant growth inhibitor. Ethylene is largely plant growth inhibitor, but it is involved in some growth promotion activities also. Plant growth regulators are involved in variety of growth, differentiation and development response. The response to the plant growth regulator may differ from one plant organ to another. Auxin promotes growth of apical bud but inhibit the growth of axillary buds. Okay. Then next is the concentration of auxin, which promotes stem growth, is inhibitory to the root growth. That means the action of the auxin is opposite in the stem and the root. Similar response may be shown by different plant growth regulators. Auxin and gibberellin promote cell growth, while the three growth promoters, that is auxin, gibberellin and cytokinin, are involved in the cell division. Plant growth regulators are effective at very low concentration, usually in the range of 10 to the power minus 6. Okay. at the site of production and the site of action, that is target cell of the plant, growth regulator may be different as in case of animal hormones, or the same unlike the animal hormone. There is no specialized tissue or gland for producing a plant growth regulator. Instead, it can be synthesized at many places by different tissue within the plant body. Now let's discuss about auxin. Auxin, in Greek, auxin means to grow. They are weakly acidic growth hormone having an unsaturated ring and are capable of promoting cell elongation, especially of shoot, more pronounced in the decapitated shoots and shoot segment at the concentration of less than 100 parts per million, which is inhibitory to the root. Among the growth regulator, auxin were the first to be discovered. However, the discovery of auxin and other type of plant growth regulators has been accidental. Charles Darwin and his son Francis Darwin in 1880 found that the senescence or sensation of unilateral illumination was picked up by the coleoptile tip of the canary grass, that is Phalaris canarensis. A decapitated coleoptile did not receive the sensation. Coleoptile tip covered by an opaque tin foil cap also could not perceive the stimulus of the light. Boysen-Jensen in 1910-13 showed that sensation of photoperiodism picked up by the coleoptile tip could be transmitted to subapical regions through a block of gelatin but not through a mica plate. Pella in 1919 replaced the previously exposed excised tip eccentrically over the stump of the coleoptile. He observed greater growth on that side even in dark. Wendt in 1928 collected the growth stimulating substance in the agar jelly. He discovered that hormone traveled by basipetally, that is from tip to apex towards the base. Agar block the containing the chemical use bending of the decapitated coleoptile according to its concentration. The growth promoting substance was named by him as auxin. Okay, the Greek word auxin means to grow. Then Kagel and Hagen Smith in 1931 isolated three chemicals from human urine. They were named as auxin A, auxin B, and heteroauxin. Kogel in 1934 found that heteroauxin is real plant auxin and is chemically indole 3 acetic acid or IAA. It is also present in urine of human beings suffering from pellagra, a disease caused by deficiency of niacin, that is nicotinic acid. Indole 3 acetic acid IAA is universal natural auxin. You can see the structure of auxin. Related chemicals are indole 3 acetaldehyde, indole 3 acetonitrile, indole 3 butyric acid IBA, phenyl acetic acid, and 4-chloroindole acetic acid. All of them have auxin like activity. Auxin is synthesized in the shoot apex, leaf primordia, and developing seeds from the amino acid tryptophan. Tryptophan help in the production of the auxin. A tryptophan independent pathway was also discovered recently. Auxin passes from the shoot tip to the region of elongation. Auxin movement is polar. It is basipetal in the stem but acropetal in the root. Auxin help in the elongation of both roots and the shoots. However, the optimum of the two is quite different. It is 10 parts per million for the stem and 0.0001 parts per million for the root. In higher concentration, auxin inhibit the growth. Raw material which is used in the synthesis of auxin is called the auxin precursor. It is tryptophan for indole acetic acid. Certain compounds inhibit action of auxin. They are called antioxin. For example, p-chlorophenoxy isobutyric acid, that is PCIB, that is p-chlorophenoxy isobutyric acid. TIBA, that is 2,3,5 triiodobenzoic acid, also act as antioxin by blocking the transport of auxin. Active form of auxin is free auxin or auxin which can be extracted easily. Auxin which cannot be extracted easily except with the help of organic solvent are called bound auxin. For example, indole acetic acid, aspartic acid, indole butyric acid, alanine, that is indole acetic acid, myoinositol, then indole acetic acid, glucan, indole acetic acid, glycoprotein. Bound oxygen is believed to be hormonally inactive, being meant for storage and protection against the degradation. Synthetic auxin. Many synthetic auxins are also being manufactured. The important one are 2,4-D, that is 2,4-dichlorophenoxyacetic acid, 2,4,5-T, that is 2,4,5-trichlorophenoxyacetic acid, IBA, that is indole 3-butyric acid, then NAA, naphthalene acetic acid, then MCPA, that is 2-methyl-4-chlorophenoxyacetic acid, dicamba, that is 2-methoxy-3,6-dichlorobenzoic acid. IBA is both natural and synthetic auxin. Synthetic auxin moves in all direction inside the plant. Now, growth root inhibitor. Okay, root growth inhibitor. The sterilized seeds of chrysis are allowed to germinate in moist filter paper. As the root reach a length of 1 centimeter or so, the root length are measured. 50% of the seedlings are placed in the test solution while the remaining are allowed to grow over moist paper. Length of the root are measured after 48 hours. It is seen that the seedling placed in the test solution show very little root growth, while the root growth is normal in the control seedlings. Now, bioassay, it is a technique of biologically activity like growth response of the substance by employing a living material like plant or the plant part. Auxin bioassay is quantitative test as it measure concentration of auxin to produce the effect and the amount of effect. First one is Avena curvature test. The test is based upon experiment of Wendt. Okay, Wendt. He did this experiment in 1928. 10-degree curvature is produced by auxin concentration of 150 milligram per liter at 25 degrees centigrade and 90% relative humidity. The test can measure auxin up to 300 milligram per liter. Auxin from a shoot tip or any other plant organ is allowed to diffuse in a standard size agar block, generally 2 into 2 into 1 millimeter. Auxin can also be dissolved directly in agar, that is 15 to 30 millimeter long oat coleoptile grown in dark is held vertically over water. 1 millimeter tip of coleoptile is removed without injuring the primary leaf. After 3 hours, a second decapitation, decapitation is carried out for the distance of 4 millimeter. Primarily, leaf is now pulled loose and agar block the supported against it to at the tip of the decapitated coleoptile. After 90 to 110 minutes, the coleoptile is found to have bent. The curvature is measured. It can also be photographed and the curvature known for shadow graph. Okay, you can see in the diagram the experiment which was performed. Now let's discuss the function of auxin. Respiration. Auxin stimulate respiration most probably by increasing availability of the respiratory substrate. Metabolism. Application of auxin has been found to enhance metabolism due to the metabolization of plant resources. Solute. Auxin increase storage of the solute inside the cell. Cell enlargement. It is most fundamental activity of the auxin. Cell enlargement is caused by the solubilization of the carbohydrate, loosening of the wall microfibrils, synthesis of more wall material, increased membrane permeability and respiration. Then Campbell activity. Degree of Campbell activity is directly proportional to the auxin concentration. Auxin also control the xylem differentiation. Cell auxin is known to promote a division of the cell of the vascular cambium. In tissue culture, the development of callus or mass of the undifferentiated cell is promoted by the auxin. Differentiation of callus occur in the presence of both auxin and the cytokinin. Auxin promote the root initiation at concentration which is inhibitory for the growth of the intact root. Apical dominance. Apical dominance is the phenomenon by which the presence of apical bud does not allow the nearby lateral bud to grow. When the apical bud is removed, the lateral bud sprout. This produces dense bushy growth. The phenomenon is widely used in tea picking and hedge making. Apical bud inhibit the growth of lateral buds by releasing auxin. It is confirmed by painting the cut end of the decapitated shoot by paste of auxin. The lateral buds remain inhibited if the apical bud is present. Now, inhibition of the abscission. Auxin delay abscission of the young leaves and fruits. Its effect is though non-functional or the non-formation of abscission zone below a leaf or the fruit. Abscission zone cuts off nutrient and water supply. However, auxin promote the abscission of the mature or old leaves and the fruits. Trophic movement. Differential distribution of indole 3 acetic acid produces tropical plant response like photoperiodism and geotrophism. Now, phototrophism and geotrophism is induced by the auxin. Now, auxin have a feminizing effect on some plants. That means it help in producing only the female flowers. The seedless fruit. The carpel producing seedless or the parthenocarpic fruit have higher internal production of the auxin that support the development of fruit, that is banana. Ethylene, higher concentration of indole acetic acid induce synthesis of the ethylene. Membrane potential, it produces a negative potential on the cell membrane. Use of auxin. Rooting. Auxin stimulate root formation on the stem cutting, that is indole butyric acid indole butyric acid aniline, and the next we have is naphthalene acetic acid. Okay, next we have parthenocarpy, application of the auxin to conjugate auxin. Okay, that is indole acetic acid and indole butyric acid are used for parthenocarpy. Okay, and other example is indole butyric acid alanine to unpollinated pistil, make them develop into the seedless fruit or the parthenocarpy. Okay, which can carry a better market price than the normal fruit having the seeds. Okay, for example tomato, as weedicide. Okay, auxin is also used as the weedicide or herbicides. They are chemical which kill the weeds growing in the field. Application of 2,4-D and 2,4,5-T removes broad leaves weeds, okay, in the cereal crop and lawn because they do not affect mature monocotyledon, while Delapon, that is called 2,2-D propionic acid, kill the grasses in the broad-leaved crops. Okay, weedicide should be used very carefully and only occasionally, as they have wide spectrum and long-lasting action. Thus, weedicides or the defoliants used in the Vietnam have exterminated the wide relatives of the citrus. Okay, then next is the flowering. The naphthalene acetic acid and the 2,4-D are often employed for inducing flowering in the lychee and pineapple. Okay, that means auxin can also induce the flowering. Storage methyl ester of the naphthalene acetic acid prevent the sprouting of the potato tuber kept in the storage. Okay, then pre-harvest fruit drop. A low concentration of 2,4-D is used in preventing the pre-harvest fruit drop of the orange and apple. Naphthalene acetic acid is similarly used for checking the fruit drop of the tomato. Vegetable crops, the chlorophenoxy propionic acid enhances the quality of vegetable crop for preventing flower formation. Next we have is fruit. Auxin enhance sweetening of the fruit, for example indole butyric acid. Okay, prevention of lodging, naphthalene acetamide. Okay, naphthalene acetamide, that is NAM, prevent lodging of or falling of the crop plant during windy season. Next is dwarf shoot. In apple, flowers and fruits are formed on the dwarf shoot. Application of naphthalene acetic acid increase the number of dwarf shoot as well as the number of the fruits.